Good evening, and welcome to our third video, Cell Communication and Enzymes. So we're going to start off with cell communication, and there are four types. We have autocrine, paracrine, juxtacrine, and endocrine. Autocrine is pretty simple. If you look here at the diagram, a cell, this particular blue cell, is releasing a signaling molecule or a ligand outside of the cell. It then comes around and binds to a receptor on that same exact cell, and that is, in essence, triggering that cell to do something itself, and that could be seen in cell growth, uncontrollable cell growth in cancer. Paracrine is the release of molecules that will act on nearby cells, where these cells are close, but they are not touching. So in this case, on the left diagram, you have a cell, and it is releasing these small molecules or these ligands that are acting on target cells that are very close, but if you note they are not touching. These target cells will have small receptors on them that are specific to that ligand. Second diagram is what you will see in the nervous system, and those are neurotransmitters and neurons. So this is what is known as the presynaptic neuron. This is what is known as the postsynaptic neuron. This area is called the synapse, and the specific space in here, in between the cells, is known as the synaptic cleft. In either of these cases, the ligand, in this case a neurotransmitter, is released and picked up by a cell very, very close. You may also see this where nerve cells are near muscle cells. Juxtacrine is our next type. Here, these cells are touching each other, and in the case of plants, we have what are known as plasmodesmata, and in animals, we will have tight junctions or gap junctions, and these are ways that cells that are adjacent to each other, that are touching, can share materials between their cytoplasms. In plants, we have these things called cytoplasmic strands, and that's the area that connects the two cytoplasms. In plants, this is important because there's a cell wall, and that cell wall is pretty tough and strong, and you may need to exchange materials, and if you can't, then cell A and cell B may have a problem. And also, plant cells do not move. Endocrine is huge in our body. Here we're going to see long-distance cell signaling, and that will happen when an endocrine gland, maybe the pituitary gland or maybe the pancreas, will release some hormone into the blood. Once it's in the blood, it can then travel throughout the entire body. What makes endocrine so effective is that this cell over here, let's call it cell A, if cell A does not have the receptor, whatever that particular ligand is or hormone does not stop there. In this case, target cell, it's called target cell B, does have the receptor. So that is how you can release these hormones over your entire body and only stop at the cells that you are supposed to. In any case of cell communication, once the ligand leaves the cell, it will bind to a receptor on another cell, which could trigger this cascade or this signal transduction that will possibly cause a cellular response. So let's look at that. In signal transduction, we have two versions that we will talk about here tonight. The first one here is called a protein hormone, and in this case, the protein hormone or the ligand is not going to go through the cell membrane. So how is the information going to get in? In this case, the ligand will kind of dock to a protein that is in the cell membrane, and this is the cell membrane. And it's a three-part story, so that's part one. It's called reception. Now, part two, many things can happen. There will probably be a second messenger. It may be cyclic AMP, or it could be a relay molecule, or it could be starting what's known as phosphorylation cascade. And any of those kind of reactions are going to amplify the original signal, and that is what happens during signal transduction, or that middle portion. In this particular example, the now activated enzyme or hormone or whatever it is could come into the nucleus. It may bind onto the DNA, which could start transcription factor. making mRNA, which will then go out to the ribosomes, which could then go on to form a protein or a hormone or an enzyme. Any number of things could happen. And that is the third part, known as cellular response. Now, what could also happen is it is possible that a second messenger, which is in the middle of the signal transduction, could be used by two different ligands. So in the left-hand case, we have a round ligand. We'll call it ligand 1. That's going to bind with what is known as receptor 1, and it's a protein receptor. And in this case, the signal can be sent. There's a second message, and it may go on to code for the square enzyme or activate the square enzyme, whatever. The triangle, which call ligand 2, which will go to receptor 2, could go to the same second messenger but have nothing to do with the rectangle and go on to make this blob-looking thing. The nice thing about second messengers and the nice things about cells are they do not waste any extra materials. If that second messenger can be used by two different pathways, well, it will. So pretty neat in the cell's case. A steroid hormone is a whole nother ballgame. A steroid hormone, the ligand itself, is going to be a nonpolar material or a nonpolar molecule. And we know that nonpolar things can go right through the cell membrane into the cytoplasm. And in this case, there still has to be reception, but this receptor is inside the cytoplasm. This is again going to be very effective. It will then go directly into the nucleus, and in this case, the transduction will be this enzyme substrate complex that has now entered the nucleus. It will bind onto the DNA. It will start transcription of mRNA, which could lead to a protein or a hormone or any other thing that could happen. Again, a three-part story: reception, transduction, and response. And we know that this will happen extremely quickly in things like anabolic steroids or HGH, which some professional athletes use. And it's, kind of against the rules for pro athletes. When we move on now, so cell signaling and cell communication is done. Now let's move our story to where the bulk of our test is going to come, and that is the enzymes. Enzymes are basically special proteins, and because they're special proteins, that means that they are composed of amino acids, and they are folded up into very specific shapes, which will have very specific functions. So because of that, they will work at certain temperatures or pHs, and those certain ranges are known as the optimal conditions, and we will see more about that. They will form a temporary enzyme substrate complex, which you can see in the picture below, and things like enzymes are not broken down. They are very reusable. So we saw enzymes last chapter in lysosomes, because lysosomes have digestive enzymes that can break down materials inside of a cell. So let's take a look at this example. This is going to be known as a catabolic example because you are actually breaking something. So in this case, your enzyme is this blue kind of molecule-looking thing. And let's say the substrate has two parts, A and B. It's bonded. They will form this temporary complex, enzyme substrate complex, and what will result are two molecules that will have been broken from one. In this case, you may also expend some energy. And this will happen in something like cell respiration, where you start off with this large molecule, let's say glucose, and you end up with these smaller molecules, water and carbon dioxide. That is a catabolic reaction. You are breaking something. We may also throw in the word exergonic, but we'll save that for a little bit later. What if it's the opposite? What if you need energy? What if it is known as an anabolic relationship or reaction? So in this case, we'll start off with our enzyme, sort of kind of looking like the letter E with these active sites. That's these areas right there. They're called active sites. And in this case, let's say X and Y are not connected. Well, we want to connect them. So it takes energy to do that. And now here is our enzyme, and it doesn't kind of looks like an E again. And now there's your X and your Y forming this substrate complex. And if you are adding energy, the enzyme stays the same. Nothing changes there. And now X and Y are linked. You just made or did an anabolic reaction. Well, there are some other ways to kind of have this active site come into play, and you will use something like a coenzyme or a cofactor. Spelled wrong here, I apologize. Let's get rid of those extra letters. A coenzyme is pretty neat. It's an organic molecule, and what it's going to do is it's going to fit into the active site, and you say, well, if something's in the active site, how is that good? But let's draw a picture. So let's say that's your enzyme, and it's kind of got a little Pac-Man looking active site. But this is the substrate. Now, if you notice, that substrate is not going to fit in that triangle very nicely. But if, let's say, a coenzyme fits right in there, taking up that extra gap, can that fit right into the now altered active site, but altered in a good way? That's what a coenzyme does. Cofactors, they're also needed in some reactions. They are inorganic, and they're going to be a metal. Now, what they may do is stabilize an enzyme. So in the case of DNA polymerase, DNA is very negatively charged, so we need to bring in a cofactor like magnesium, which is positively charged, and that will chill out the reaction and stabilize it. So if we take a look at this picture over here, the inorganic piece that may be sitting over here, that's your cofactor, and that again, once again, could be a magnesium, let's just say. And if that cofactor is not there, the enzyme may be inactive or work very, very, very slowly. In this case, the coenzyme looks like it's hogging up the entire active site of this enzyme, but it's actually not, because the substrate, if you look, kind of looks like a block, if you will, and that will fit right in here. So everyone's nice and happy. So coenzymes and cofactors are very, very, very necessary. And once again, if they are removed, if the cofactor is removed, so if removed, it is possible that the enzyme will not function. Oh, what happened? Let's bring it back. Okay. So, also, one thing I forgot to mention before, that those anabolic reactions and coenzymes and cofactors and all that happens in plants and in animals. So both plants and animals. So let's not forget that. Very important. Let's make sure we're still recording here. And we are, so I guess we can kind of go like that. Yep, all right, we're having a little trouble with this evening. All right, let's see. Let's move our story down. Now, we already said before that if you put something in the active site, if it's a coenzyme, it's good. But what if it's not good? That is known as competitive inhibition. So in this particular example, you can see that the enzyme is this purple-looking goldfish thing. And in the event of something what is known as a competitive inhibitor, now that is blocking the active site. So as the substrate tries to get in, well, it can't. So what that may do is slow metabolism. And again, metabolism could be any chemical reaction that's going to be slowed. And in this case, the enzyme, it can't do its job because it's being blocked. Another way to kind of mess up the active site is to have what is known as a non-competitive or allosteric reaction, inhibition. And in this case, the allosteric or non-competitive is not in the active site, it's over here, but it alters the shape of the active site so the substrate does not fit. And again, this will also slow metabolism. So all of these examples where we mess up the active site are going to be negative for the enzyme and substrate and the act of trying to break them down. So now, how does all this, what's another way to kind of inhibit things? And again, inhibition means to stop or slow or mess up. So in this case, I have three enzymes and four kind of spots in the story. We may see something called feedback inhibition. And what will happen is A goes to B with the help of enzyme 1, B goes to C with enzyme 2, and C goes to D with enzyme 3. And if the end result is to make whatever substance D is, that's great. Well, what happens if you have enough of D? Well, the cell's not going to keep on making more if you don't need it. So what will happen is this D may go back and inhibit either enzyme 1 or it could be enzyme 2 or enzyme 3. In this case, it happens to be enzyme 1. And what happens when it inhibits enzyme 1, you will get no more products here, and that's fine. Once the D is gone, then since there's no D, enzyme 1 is no longer inhibited. The reaction continues until the cell makes enough D, and this system shuts off and begins. And it's brilliant because, again, cells do not want to waste energy. So in this example here, D shuts off enzyme 1, and since there's no enzyme 1, the whole thing is going to shut down. Well, what about enzymatic reactions as something going on? Well, we measured this in the lab. We measured the production of product, and we did catalase, which is the enzyme, up against the substrate hydrogen peroxide, which is H2O2. Now, you can measure two things in reaction, the loss of substrate or the production of the product. And, well, the substrate's clear, so it's kind of hard to measure. But we were able to measure the product, which was oxygen production or oxygen bubbles. So that was pretty cool how we did that. Okay, and sorry. Now, how could we increase the rate of a reaction? Well, some of the lab groups were adding salt, and some lab groups were changing the pH, and that messed up the reaction rate. But let's say I want to keep it simple, and I have an enzyme that's doing a reaction at whatever rate it is. The way I can increase the rate is I can add more substrate. That's okay. But the real limiting factor is probably going to be the amount of enzyme. So if I want to speed things up, I add more enzyme. So if we look at we'll use our blue line in the middle for the kind of benchmark, I guess. If we add more enzyme, then we jump up to the red line. If we have less enzyme, then we fall down to the yellow line. And that should make sense. And you can see here in this area that A and B are the same exact rate as each other. But does that line have a faster rate than both C or D's line? And that's the key. So by adding more enzyme, you speed up the rate and you will make or digest more of the substrate. But if you notice, though, all of the lines will eventually level off. And the reason why they will all eventually level off is because all of the substrate has been used up or digested or broken down, however you want to say it. And what if I had no enzyme at all? Would that be this line down here? And that's what happened when we tried to trick everybody by giving you just water and see what the enzyme did to that. So if there's no substrate or there's no enzyme, you have no reaction. So what can be said for the first several minutes of, let's say, A to B? Since I wasn't very clear on my instructions, we know that that is a constant rate. And when the graph flattened out, either all the enzyme combined with the substrate, or there was no more substrate, or the substrate was consumed. That's another way you can say it. Substrate was consumed. Well, what about optimal temperature and optimal pH? In this example on the left, the optimal pH for the red line is about 2, maybe 2.5. This may happen in your stomach. And in the blue line, it's about 9. That may happen in your intestines. And it just goes to show that different sections of your body have different pHs, and the enzymes there will work at their own specific conditions. Temperature, this may be our body, somewhere around 37 degrees. Way up around high 70s, and again, this is in Celsius. This could be the bacteria that may be found in hot springs. And when they found all of these organisms living in these hot springs or these ridge vents, which are at the divergent boundaries in earth science, if you remember, it's hot. So therefore, their enzymes have to be able to handle that temperature difference. And then how does the location of an organism determine its enzymes' optimal temperature pH? And again, same thing. If you live in polar oceans, well, your enzymes will prefer lower temperatures. If you live in a hot spring or thermal vents, you will have high temperatures. If you are in places like your stomach, you will work best at a low pH. So it all depends on where the enzyme is and where the substrates are and where in the body you are. And All that, pretty important. Gibbs free energy, which is our last piece of the story, which will kind of bring into everything we've done. Gibbs free energy is a formula where you have G equals delta H minus temperature slash delta, if multiplied by delta S. Each of these has a meaning, so free energy is G. Enthalpy, which is the total energy of the system, which is your H. Temperature is in Kelvin, and entropy is your amount of disorder. So if you are breaking up a big molecule, let's say you're going glucose to its broken down pieces, which could be H2O and CO2, then that is going to have a increased delta S. And if you increase your delta S, meaning you make this bigger, you're going to have a larger minus number, and your overall G will be negative. If you are taking small pieces and you are building up a larger molecule, let's say you are taking CO2 and water, and you are building glucose, in this case, you are decreasing your amount of disorder, building of molecules, and here you will have a small delta S, which means you will have a small negative, and your G will be a positive number. So how does that all play? Well, what does a negative G mean? A negative G means that the reactants have a higher energy than the products. This is said to be spontaneous. It will break the bond into smaller molecules. It is known as an exergonic reaction, and cellular respiration is a great example where that expends out ATP. So if you look here at this graph, you will see the reactants have a certain energy level. The products is lower than that. So when that happens, it is known as a spontaneous exergonic reaction, where your delta G is less than 0 or negative. Now you notice there's this little bump here. This little bump is called activation energy. And even though the products are lower than the reactants, you still have to get over this bump. Now, in our bodies, heat is enough to do so. But in the event that heat is not available, or you just want to make things quicker, you can lower your activation energy with the help of a catalyst. If you notice this green line here, you did not change the energy of your reactants, you did not change the energy of your products, and you did not change your overall G. You just made it faster. So what is activation energy? And that is the energy that is going to kind of destabilize the bonds. Let's say it's glucose breaking up into water and carbon dioxide. If you want to speed that process up, once again, you add a catalyst, and the catalyst is that green line there. And we are not adding a ton of energy to get this reaction to go. You'll see that later. And again, by adding the catalyst has no effect on your total energy drop. And finally, let's answer this last question up here. How does the catalyst affect the overall free energy? And we just said there is no change. The exact opposite of all of that is if you have a positive G value, or you have what is known as an endergonic reaction. This is not spontaneous. You need a ton of energy. And if this is photosynthesis, you are getting your ton of energy from the sun. In this case, you have to climb this very large mountain, and the only way to do that is by having energy added, in this case from the sun. Now, inside of your body, you may have this endergonic reaction that you need to couple with, let's say, ATP hydrolysis. That's going to give you a few calories, about 7. The negative G is about 7, so that can help you. But in the case of photosynthesis, there is no way in a million years that's going to happen. You need to get energy from something, in this case, the sun. And yeah, there's still a little activation energy needed, but it's not the same. So hopefully all of this will help, and good luck on your test.